Well, welcome everyone. Bienvenue tout le monde. Wea. I'm so happy to be here with you all, sharing this day with you. My name is Tina Semeniuk. For those of you who don't know me by now, I'm the director of FishCAST, which is a CREATE training program that is meant to train the next generation of scientists and practitioners in the field of conservation of and management of fisheries. And I am so delighted that our seminar series for this year is all about the FishCAST trainees that have been through the program and come out on the other side. And we just love to just uphold their voices and support them as much as we can one's not only when they're in the program, but when they're out of the program. I am coming to you today from the University of Windsor, also known as Wawaetanong. That's the Anishinaabe term for where the river bends. And I'm on the Three Fires Confederacy of the First Nations of the Odawa, the Ojibwa, and the Potawatomi. And I find myself so privileged to be able to work here in this region, which is so high biodiversity, but is a region that's known as an ecoregion of crisis. And so, again, I feel so privileged to work here and to work with all of you, whether or not we're doing it virtually, remotely, and the partnerships that we've all collated and created together just really inspires me and just I don't know, keeps me going. You all keep me going. So I'm so pleased to be here. But I'm pleased to also have been able to know Brooklynne Keber, formerly at Litke. She comes from Dr. Margaret Docker's lab, and I'm not going to do too much of an introduction because I'm going to leave that to Margaret, but I do want to say that the interactions that I've always had with Brooklynne have just been just so wonderful, so positive. Brooklynne's always been such a keen collaborator, always getting involved. Brooklynne, you've never minded to be vulnerable and to share your feelings and your thoughts and your opinions. And as a consequence, I felt like not only did I learn more about you, but I learned more about everything that all of our trainees encounter and touch upon. And so I wanted to thank you for being part of FishCast. I wanted to thank you for the learning that you shared with me. And I'm just so pleased just to learn about where you are now. And with that, I'm just going to turn the bulk of that over to Margaret. But I just wanted to say welcome back, and I can't wait to hear where you're going next. Thanks. Merci, miigwech.
Okay, and I apologize if you can hear the whirring sound of my computer, and I hope it doesn't die as during the next hour. Yeah, it's just such a pleasure to introduce Brooklynne. Brooklynne finished her master's in my lab at the University of Manitoba this past summer, but she's been an essential part of the lab since what was the spring summer of 2020, during the upheaval of the pandemic, when she decided that fish and eDNA research is what she was interested in, and she sort of threw herself into it full force, doing simultaneously the pre-honors course with a lit review and proposal on eDNA, reading seminar-based course on using eDNA to monitor aquatic organisms, all while also starting her honors project using eDNA to map the distribution of chestnut lamprey in Manitoba. And as I said, she's just been an integral part of all of the work in our lab since then. And it's a great pleasure that her finishing her work doesn't sort of end our association, but with her new job as a conservation and research specialist at the Assiniboine Park Zoo, that we'll be able to continue working together. I'm hoping for some time into the future, although I suspect it'll be more the fins side of thing than the fur side of things. But with that, I'll let Brooklynne tell you all about her eDNA journey.
Thank you both so much. It really warms my heart, and you guys are getting me a little bit choked up before starting this. As Tina and Margaret mentioned, my name is Brooklynne, and I'm both a FishCAST alum and Docker Lab alum. And today I'm going to be talking a little bit about my journey from working in fisheries or fisheries-based research to the wilder and broader taxa that I now work with at the Assiniboine Park Conservancy as a conservation research specialist. So how it started was, as Jessica Reed was talking about last week, as a naturalist as a child, I was interested in the natural world. I grew up as a farmer's daughter. My dad grew up as a kid on a farm, and my grandma still lived on that farm. And my dad took over the farm when my grandma no longer could take care of the farm. So I grew up on the farm and was always outdoors, luckily. Had that privilege to be able to spend so much time outdoors and be able to see so many interesting things. And because of my interest in the natural world and all of the things that come with it, I started my route towards pre-med because as Jessica alluded to last week, when you're a kid who likes biology and things like that, there seems to be kind of a natural progression to at least start going into pre-med when you go into your university career. And I really enjoyed that for the first little while. I worked in a hospital for a good portion of a couple of years. And while I did enjoy the diagnostic side of things, the patient care was not exactly what I was looking for. Research really intrigued me. I enjoyed my labs and especially my microbiology labs, but the idea of spending the majority of my job with patients and the charting and patient care side of things wasn't really it for me. So I transitioned to a Bachelor of Science degree looking at veterinarian medicine. At that time, I was spending a lot of time personally with my dog, going to the vet. He is what I'd like to call a bit of a lemon. So we got to see a lot of different sides of veterinarian medicine. And at that point, I thought that veterinarian medicine was the right thing for me. I was working with rescues on my spare time, helping with dog transports from northern Manitoba to different areas of Canada. I was also going to the zoo where I now work to do some animal behavior studies with courses. And it seemed like the animal side of things was a better fit for me. But with animals or non-human animals still come humans. And I realized again, it wasn't the right fit for me. And then around that time, I have a picture of me somewhere holding a lamprey, but I couldn't find it for the life of me. I ended up taking Dr. Margaret Docker's course, the core dates, and something clicked. I saw Dr. Docker's passion for what she did, her research, lampreys, and I just knew that whatever she had, whatever was her formula was what I wanted. And I stuck to her like glue, and I just knew that I wanted that too. Whether it was lampreys or not, I knew that she'd have the answers for me to be able to get to what I wanted. And lucky enough, for me, I was right. So I transitioned to a Bachelor of Science honors degree and started my career in research. As Margaret mentioned, I started my honors project or my honors thesis investigating the use of environmental DNA to map the distribution of chestnut lamprey in Manitoba. And because I believe the majority of you are in the Great Lakes region, I'm sure you know what lampreys are, but in case you don't, they are jawless fishes that are present here in Manitoba. You might be familiar with sea lampreys, which are not here in Manitoba. They're the kind of bad characters in the lamprey world. But the majority or the rest of the lampreys in Canada and all the lampreys in Manitoba are non-invasive lampreys and are beneficial to the environment. So looking at the good guys, and especially the chestnut lamprey, they are considered data deficient by the Committee on the Status of Endangered Wildlife in Canada, CSIWC, and were listed as such in 2010. And the reason being is a lack of information regarding their distribution and abundance in Canada. So with this project, I wanted to fill the knowledge gaps regarding their distribution in Manitoba, and I wanted to do this using environmental DNA. So as Margaret mentioned, it was COVID, and it's also an honors project, so it made a short timeline a little bit shorter and a little bit more difficult. So my sampling efforts were small, and what I was able to do was small as well. And I'll get to that in a little bit, but to give a background on environmental DNA, if you're not familiar with it, environmental DNA or eDNA is just DNA that's extracted from the environment. So it can be from anything that's shed or excreted from an organism, and it's in the environment and contains DNA. So the difference between a typical DNA sample versus environmental DNA sample is you're taking the DNA from the environment rather than the organism itself. And the environmental DNA samples that you collect in environmental DNA in general can be affected by many different aspects or characteristics of an environment. So because I was looking at fish and I'm collecting environmental DNA samples from an aquatic organism, the water temperature, the pH of the water, the presence and absence of microbes and enzymes in the water, inhibitors in the water, and the time in which the DNA was shed and how long it's been there can all affect whether or not I'm able to detect the DNA in my sample. The process of eDNA sample analyses, or just the general workflow for environmental DNA looks like this. So first you collect your sample from the environment, then you filter your sample to isolate the environmental DNA from within your sample onto a filter, and then you take that isolated environmental DNA and extract it from the filter into a pure aliquot of environmental DNA, and then you can take that aliquot of environmental DNA and analyze it using a various method of polymerase chain reaction, or PCR. I used quantitative PCR or qPCR to tell whether or not your target species is, or the eDNA of your target species is present in the sample. So as I was alluding to earlier, I had a minimal sampling effort because of the honors project, COVID and whatnot. So I wasn't able to go to many places, but I was able to sample a decent amount of samples within the known range. But unfortunately, I was only able to detect chestnut lamprey eDNA within one of the samples of the samples I collected. I was using an assay that was designed by someone else, and it was a bit of an older assay in its design method. And I had suspected that potentially that could have been one of the reasons why I wasn't able to detect chestnut lamprey, because a lot of the samples that I collected were within their previously known range. So either there could be some severe range restriction or extirpation of chestnut lamprey, or maybe something else is going on here. So I was lucky enough to stay in the Docker lab through my FishCAST trainee program as an undergraduate, and continued in 2021 in the Docker lab. And I eventually designed a genus-specific assay for native lampreys in Manitoba that targeted chestnut lamprey, northern brook lamprey, and silver lamprey, all of the native lampreys here in Manitoba, which are all within the genus Ichthyomyzon. And I was able to detect Ichthyomyzon eDNA in many more samples than what I detected in my honors project with the older assay. So that kind of lit a fire in me and inspired my master's thesis, if you will. And I continued on in the Docker lab with my master's thesis in 2021. So it started off with Manitoban native lampreys, the chestnut, northern brook, and silver lampreys. And I love this photo here because it also includes the sea lamprey, which is the big guy. Get my pointer here, the big guy here. So when we think about the good versus the bad lamprey, the destruction that sea lamprey can cause, you can kind of see why that might be, considering the differences in sizes and why these smaller lampreys are not as destructive as the sea lamprey. Anyhow, so my master's thesis started tracking freshwater fishes of conservation concern in Manitoba using environmental DNA, just focusing on these native lampreys. And as master's projects go, I added on, if it will, doesn't want to. That's all right. Okay. Next, there you go, bigmouth buffalo, which is a sucker species here in Manitoba. Then golden red horse, another sucker species, and horny head chub. And you might be wondering why. And I think I was too, and my thesis committee certainly was as well. But what I could kind of come up with, and to summarize these, is these were kind of the underappreciated or the underdogs in Manitoba. These were the fishes that were often overlooked. Suckers, minnows, and lampreys. No one seems to really care about these fishes as much. They don't get the same kind of attention as some of the more charismatic species. So these under-charismatic or less charismatic species that are still incredibly important to the ecosystem kind of became my passion. Bigmouth buffalo is having its moment, I will say, but I started targeting these species as my interest, and they all are of various levels of conservation concern, and they could all benefit from being tracked using environmental DNA, and all of their suspected ranges did overlap. So there was some method to my madness. So from 2021 to 2023, I sampled extensively across Manitoba. I sampled 53 sites 4 times, either in May and June, July, August, or September in 2021, to see if there was any sort of temporal difference in the detection that I would get, and I didn't notice anything significant. So then in 2023, I sampled 69 sites, focusing more on range expansion for sampling. In my master's project, I was able to design, validate, and use TaqMan MGB probe-based eDNA assays to detect bigmouth buffalo, chestnut lamprey, golden red horse, horny head chub, and northern brook and silver lampreys in Manitoba. Using those assays, I mapped their current distribution and also compared eDNA sampling to capture-based sampling. So with this incredible sampling effort, I was able to see Manitoba in a way that I'd never seen before. I was able to go sampling in all these fantastic lakes and rivers within Manitoba. I was able to see fishes in during spawning. You can see horny head chub, common shiner, carmine shiner in here in that video there. And also do it with one of my best friends of over a decade, who now happens to work in the Riverine Ecology Lab, which we've collaborated with extensively, which was pretty fantastic. This sampling also took me up to Churchill in Manitoba, where I had that special feeling I had again that I had when I first met Dr. Docker. And I realized that Churchill was a special place for me. And whatever I did in my career, I had to have some sort of link to Churchill. So graduation came, and I want to specifically mention that the work that I did in my honors and my master's would have not been possible without many, many people, and of course the support from many agencies. So I just wanted to take a brief moment to thank the people mentioned here, but also many others that I couldn't take the time to mention here. It is, these projects are so collaborative and include so many people, and I just am so incredibly grateful for all the support that I had throughout those projects. But with graduation came, and I was looking for a job, or a little bit before graduation, I was looking for a job, and I specifically knew that Churchill was something important for me, and that I wanted to stay in Manitoba as well. And I was lucky enough to be able to join the conservation and research team at Assiniboine Park. So specifically, I would like to mention that, give me a second here, sorry. Oh, and the Assiniboine Park is located on Treaty 1 territory within the province of Manitoba, and is on the traditional lands of the Anishinaabe, Cree, Oji-Cree, Dene, and Dakota people, and the national homeland of the Red River Métis. So all of the work that we do here at the Assiniboine Park Conservancy is within Treaty 1 territory, and we, of course, like to take strides in moving past that just statement of land acknowledgement and work towards reconciliation, which I'll chat about in just a bit. So as I joined this team, I joined a bunch of fantastic and amazing conservation and research scientists, and joined a different kind of style of research than what I was used to. So Assiniboine Park Conservancy is a non-for-profit, and all of my research thus far had been in an academic setting. Specifically, the Assiniboine Park Zoo is an AZA accredited zoo. So the Association of Zoo and Aquarium require 4 specific criteria to be met for accreditation, which it's more complicated, but just to summarize it, you need to adhere to sustainability and green practices, field conservation efforts, conservation-related research, and conservation education and advocacy. Of course, our department contributes partially with the conservation education and advocacy, but substantially towards the field conservation efforts and conservation-related research. Our conservation themes fit within 4 major themes: understanding the changing Arctic, preventing species extinction, protecting urban biodiversity, and cultivating conservation connections. So just giving some brief summaries of some of the work that I'm doing now, from that transitioning from fins to fur. Never mind. Let's go back. And I click on you. Okay, we're just gonna have to not do that one today. Of course, are some of our ambassador species, the polar bears. So the polar bears in the wild that we head up to Churchill to research. Specifically, we provide tools to help manage polar bears and their habitats. Right now, we are looking at completing a map-based resource for land use and fire risk mitigation that help both with protecting denning locations and advising about potential fire risks for the Churchill region. For another species up in the Arctic region, we look at Arctic harbor seals. Specifically, we want to understand the movement of and the river use of these seals in the Western Hudson Bay. So the team travels up to Churchill in May and outfits the harbor seals with satellite transmitters. This year was one of our most successful years to date, and 6 adult harbor seals were outfitted with satellite transmitters. So this satellite data here can shows some of these movements of the seals within the Churchill River and the Seal River over here, and then also the Hudson Bay, which is a marine environment. Beluga Bits is one of our other Arctic projects, and the goal with this project is to collect underwater footage and above water images of beluga in the Churchill River. It is a community science project hosted on Zooniverse, and if you're interested in joining the project, you can scan the QR code to join and help us to identify the age, the sex, the size, and other characteristics of the beluga in the Western Hudson Bay population. So our team heads up to Churchill in July to collect these photos, and during that period, they collect hundreds of thousands of photos to create this database to help with understanding the population of beluga in this region. And part of that cultivating conservation connections, we are part of a team that facilitates conservation leadership. We participate in conservation assessments and recovery planning with CACIWIC, and we work with other organizations for conservation planning, such as the Saving Animals from Extinction SAFE program through the Association of Zoos and Aquariums, and the Species Survival Commission through IUCN. One of our preventing species extinction programs includes the Grassland Butterfly Conservation Program. One specifically is the Poweshiek and Dakota skipper grassland conservation. These are two grassland butterflies that are listed as endangered by Kasiwik that are found here in Manitoba. The decline of these species is largely unknown, but is probably in part due to habitat loss. The Poweshiek uses tall grass prairie, which is an endangered ecosystem. Less than 1% is remaining in Canada. And what we do here is head start the individuals at the zoo. So we provide them with a safe environment to grow during the most vulnerable stages of their life. And then once they become adults, we release them into the wild to produce the next generation. So this has been a incredibly successful program, and we've seen record releases year after year. And these numbers have been incredible, especially considering that for the Poweshiek skipperling, in 2018, before the program started, there were less than 100 individuals estimated in the wild. And just last year in 2023, we released over 400. Manitoba Mud Puppy is my baby project now. So it's one of the projects I just started recently here at the zoo, where I'm able to take the skills and expertise that I gained throughout my honors and master's thesis and expand it onto a different species. So Mud Puppy in Manitoba are data deficient, and they have received a status recommendation of threatened by Kasiwik. And so the goal of this project is to fill the gaps in knowledge for Manitoba Mud Puppy using environmental DNA sampling, trapping, and habitats suitability sampling. So this project just started a couple months ago, so we're still in the midst of it, but it is nice to get back to my roots. Within the zoo, we protect urban biodiversity by providing safe habitats for aerial insectivores such as chimney swifts, tree swallows, purple martin, but we also mitigate window strikes on all the windows, as many window surfaces as we can at the zoo. We have a lot of windows throughout the zoo and on various buildings in the park. So by mitigating these window strikes, we try to minimize the impact on these aerial insectivores because they have been noted to have some pretty dramatic declines in populations, with the collisions with windows being one of the most significant causes of these. So these nest boxes and providing habitats for them to reproduce in have been one of the successful ways that we've been able to help these populations. The artificial chimney that you see here has been was one of the first ones, or the first one that was installed in Manitoba, and we've had successful fledglings from it every year since. And us as a department, we've been making active efforts towards reconciliation and capacity building by supporting Indigenous conservation initiatives, including bison rewilding projects with First Nations and creating opportunities to build capacity. We've been working actively with Seal River Watershed to create an Indigenous protected area. And part of the harbor seal work that we have been working with them to accomplish has been to identify where these areas are that should be protected. One of the amazing parts of my job now is that I get to be part of training future conservation scientists. So at the zoo, we also support students and visiting researchers, and are always creating space for more training opportunities. So over time, from my research as a student to now my research as a conservation research specialist, some stayed the same. So this is me just last month in the field with Dr. Stefan Peterson, the director of our department, collecting some eDNA samples at the Winnipeg River, which is a site I've visited almost every year now. And a lot has changed as well. I do genomics work Powashik, looking at inbreeding and genetic diversity, looking at gene flow in the tallgrass prairie preserve, looking at whether we should attempt assisting gene flow and genetic rescue. Is there inbreeding depression? Can we detect it? Is there a correlation between high genomic inbreeding and reduced fitness? I'm also looking at population structure and gene flow and cougars as they recolonize in Western and Eastern Canada. Cougars in Manitoba are present, but there doesn't seem to be a breeding population. So we're wondering where these cougars are coming from. Seems to be that they are coming from a mixture of North Dakota and also Saskatchewan, Alberta, but more information coming soon. But to summarize all of it, what I've learned over my time as a student and now as a person in their career over the past, I guess, 9 months, 6 months, something like that, that I'd like to impart on the next generation is to learn to market yourself and identify the skills you've learned. Soft skills are just as important as your hard skills, and break those larger skills that you do have into smaller skills that are transferable. For example, to be able to do PCR, you need to be able to do precision pipetting. You need to be able to know how to have clean technique, electrophoresis, prepare an egg roast gel. And these are all things that you can then apply to different research projects. It's not necessarily that you're going to always be pipetting on a PCR gel. It's you might be pipetting to do something else. So having these skills can apply to other projects, and that's what's going to be important as you transition into a different career, and what's helped me from going to doing eDNA for 4 years to then working on so many different projects. Another thing is identifying what sets you apart from others. Everyone is unique, and I'm sure, as all you know, being in the FishCAST program and meeting other trainees, is you're all doing different projects, but you're all different individuals. But even your hobbies can help set you apart and help find you a career as well. And those hobbies can be beneficial as well. So things like, if you're a lifeguard, do you work on cars as a hobby? Do you have a boating license? Do you have a firearms license? These are all things that can, in their own way, also set you apart from other candidates for jobs. Firearms license are important for when you're working in the Arctic. Having a boating license can be beneficial for if you're going to be doing fisheries works. If you work on cars and are familiar with motors, that can really be beneficial if you are working on boats, and because boat motors tend to stop working a lot of the time. And being really comfortable on water by being a lifeguard can also be really beneficial working in fisheries. But the most important thing that I found out throughout my years and continue to learn every day is how important networking is. Not only networking, but making meaningful connections. I'm really grateful for what Tina said at the beginning of this presentation, but it does really feed into it, is that making those meaningful connections and spending the time to participate and ask questions when you are meeting people really makes a difference, and can lead to either collaborations in the future or an opportunity for you to ask for help for someone in the future. Especially now that I've come into a position that I'm outside of my comfort realm, I've had to lean on many of my colleagues and people that I've reached out to in the past or were connected with in the past, and having those connections has been so important for me. Going back on my FishCAST connections has been important for me, and going back to the stuff that I learned through in my career credentials and having made those meaningful connections, learning from others, has really been incredibly important. The job that I have now, or the career that I'm in, started in part because of some networking that happened in 2 different occasions. I first met my boss at a conference, and a couple times. Is it the second time? I honestly don't remember now, but I met him twice before I ended up applying for the position. And I knew about the job that I ended up applying for, because I ended up hearing about it coming on the market, I guess, through networking, and I had the career alerts on for the Assiniboine Park Conservancy because of that. So I really, if I can impart anything to you, it's the importance of networking and really stepping outside of your comfort zone to try to make those meaningful connections. So thank you so much for giving me the opportunity to share my experience and how I've come from a FishCAST trainee to the person I am today, and continue to learn from this point onward. My contact information is there, and I'm more than happy to answer any questions through email if anyone is more comfortable in that form. But otherwise, I'm happy to take any questions now as well.
Brooklynne, thank you so much. That was amazing. And I can't tell you how happy I am to hear that you're working up in Churchill, that it gave you those warm and fuzzies and you're able to pursue that. Like, that's just incredible. So I'm glad you felt that with Margaret and you capitalized on it. And then you felt it again in Churchill and you capitalized on it. So that was amazing. Margaret, I'm happy to turn it over to you to moderate for the next little bit, and just having to let the audience know that we'll open up for general questions, but then I think Margaret, Kendra, and I will leave you all so you could ask more personal questions to Brooklynne that you want, if you don't want to do it in front of us, but we want to have you set up your own networking without the specter of us looming over you. So Nina said, thanks for the presentation. It was great. And